Hello, in this video we will discuss about the cell plasma membrane and we will also understand about the structure of cell and acts as a semi-permeable membrane and physical structure of cell and function of the cell we will discuss in this video. So first of all, we should understand about the concept of the cell in the plant and animal. And what is the cell? Cell is the basic structural and functional unit of life of the plant as animal. These are the multicellular, but the unicellular also amoeba or paramecium is also a unicellular eukaryotes. But on the other hand, the bacteria is the prokaryotes unicellular, which that is lack of nucleus. But let's here we will discuss about just the plant and animal cell, and next we will discuss about the plasma membrane and their structure and function. So first of all, here is the plant cell. Plant cell contain a different as compared to the animal because it contain chloroplast and cell wall. So what is the importance of this? This importance make a autotrophic cell. So the autotrophic cell means it produce energy with own self by the using of light in the presence of chlorophyll in the present into the chloroplast, and the carbon dioxide used to produce the glucose. So that is known as the autotrophs. Auto mean itself, trophic mean the produce of energy in the cell. So that means the paramecium or other euglena, which that is also contain a chloroplast or something organelle, which that is used for the phototactic movement, negative and positive, etc. But here is the cell wall. We will discuss about the cell wall, and vacuole are present bigger as compared to the animal cell, and vacuole used for the storage of water and solution. But cytoplasm is present and plasma membrane is present also, same a human being or animal. So here is the black color shows that the symplast pathway occur in the cell wall to cell wall junction, while on the other hand the symplast to apoplast pathway is used for the cytoplasm to cytoplasm pathway for the movement of water, while the vacuolar pathway is also occur in this mechanism. So cell wall is made up of cellulose. So the cellulose is used for the absorption of water and imbibition. So let's begin to understand the animal cell. So animal, here I am drawing the person which that is contain a tissue. This tissue is the basically heterotrophic tissue. So the heterotrophic cell contain a just plasma membrane and not cell wall. But on the other hand, lack of chloroplast, so it means it's consume energy from the plant and other sources. So it means it is known as the heterotrophs because it is not produce energy itself by using of light. So here I am drawing the plasma membrane which that contain phospholipids, phospholipids, bilayer of phospholipids are present, which that contain basically here is a phosphate group are attached is hydrophilic polar hydrophilic, but hydrophobic is the fatty acid chain, which that is unsaturated fatty acid is basically contain a double bond and it bend their chain, but though two tail are present and one is the saturated and one is the unsaturated, because the saturation is the meaning of that the double bond are not present in the fatty acid chain, while on the other hand the double bond are present, which that is oil like structure. So the saturated and unsaturated fatty acid give a play important role for the fluidity we will discuss in the next. But first of all we should understand here is the phosphate group which that is attached with the polar molecule. For example, water can attach, but it will not move inside and it will not move outside. So due to the inside is the hydrophobic interaction, but on the other hand the outside is the hydrophilic. So here is the steroid is the non-polar and it is easily passed into the lipid soluble because it is lipid soluble and pass into the inside of the cytoplasm. So it is easily passed due to the hydrophobic lipophilic and hydrophobic interaction. So that means the lipophilic means the inside is the lipid fatty acid which that is allowed to move inside the steroid, and basically we will discuss also what is that. And so here is the receptor of cell which that is basically epinephrine, glucagon, insulin signaling molecule which that will not pass into the because it is bigger molecule, and receptor of cell are present is here, and these receptors contain a peripheral protein. For example, G couple receptor is present, so G protein are present, which that is peripheral protein is not completely covalently bond and dissociate during the primary messenger signal, for example epinephrine, glucagon, insulin signaling molecule, which that give the stimulation by the using of ATP to convert phosphate phosphorylase G protein, which that is go into the adenylate cyclase enzyme, which that is present also protein embedded into the plasma membrane is known as the integral protein. So it means the ATP use, and ATP used to produce 2-phosphate group released by the adenylate cyclase to produce cyclic AMP. So this cyclic AMP will be second messenger. So this second messenger is basically this is the signaling pathway, which that is the secondary messenger signaling pathway. And on the other hand, here is the basically ATP used by the cytoplasmic protein to activate with the attachment of phosphate group by a secondary messenger. So it will be go into the transcriptional factor, specific transcriptional factor on the basis of signal to transcribe the DNA into the messenger RNA. So this is known as the transcription. After this transcription, the messenger RNA will go into from the nucleus to the outside of the cytoplasm as a messenger RNA. So this messenger RNA give the protein by the translation, and that means it is known as the translation. So that protein is the specific for every purpose. So remember about that. If the myosin or actin is preparation in our muscle, so it means this protein can be a myosin and actin, which that is play functional role. But on the other hand, next here we will discuss about the transport. So the transport into the cell, outside and inside of the movement of the molecule, we will discuss in this different mechanism. So the different mechanism can be possible. But first of all, we should understand about the asymmetrical phospholipid bilayer. So the phospholipid bilayer is in the form of asymmetrical, not asymmetrical. Why? Because upper phospholipid layer is contain a different molecules as compared to the inside. So the upper layer is contain a phosphatidylcholine as well as phosphosphingomyelin. On the other hand, here is the downside. Inside is a phosphatidylethanolamine and phosphatidylserine, which that is make the difference inside as compared to the outside. So carbohydrate is also present, antigen is also present. Antigen is made up of glycoprotein, which that is specific, and the viral infection can be triggered by this antigen, or you can say that this antigen is basically used for, against the basically our immune system will give the again these produce antibodies. So the carbohydrate is also present. So let's begin to understand. Here is the enzyme also is the integral protein. So this enzyme integral protein are present in the epithelial cell of the digestive system, contains sucrase, peptidase, dipeptidase, etc. So that's used for the breaking down of smaller molecule into more smaller. So let's begin to understand about the transport. So active transport and passive transport is occurring in the cell via a different mechanism. Active transport is used for the ATP, while the passive transport is used without the energy consuming process. So let's begin to understand how it's possible. So this was the passive transport. For example, inside is the water less, so outside it will be more. So it means the porine is contain a protein, integral protein channel, which that is allowed to move the water inside. Due to the toward the osmotic gradient, it means the inside is the less and outside is the more. It means Requirement of water inside is more, so that means the semi-permeability of this membrane will allow to move the water inside from the outside. So let's begin to understand the active transport. So active transport use ATP against the osmotic gradient. For example, inside the potassium concentration is more and outside is the potassium sodium concentration is more, but it will be move against this osmotic gradient. It means against this concentration. For example, outside is the less potassium, but resistance to move inside due to the using of ATP to change their electrical charge. For example, the neuron, and for example, the muscle contraction, or on the other hand, for example, the movement of sugar inside of the cell by using of ATP. So I hope you make sense about these things. Let's begin to understand osmosis. Osmosis is basically is the directly semi-permeability membrane which are used to water move inside. So that means the osmosis is basically the movement of any substance against by the semi-permeable membrane, just like a passive transport, basically the osmosis. So here is the diffusion. What is diffusion? Diffusion means the very small molecule, gaseous exchange is occurring in the cell. For example, the oxygen will move into the mitochondria from the outside extracellular fluid to the inside of the cell, and carbon dioxide released to produce glucose to convert pyruvate, and this pyruvate move into the mitochondria and produce ATP by production of acetyl-CoA in the mitochondria. Mitochondria is a double membrane organelle, so remember about that. So the next we will discuss about the cholesterol. Cholesterol is the also important part of the cell membrane. So the cholesterol is embedded and it's contain a polar side also, OH negative. So what that means? It means the fluid retention will be occur, fluidity will be occur. Why? Because water is attached with the OH negative hydroxyl ion. So the interaction in between the phospholipids also. So it provide the gap or provide the space due to the embedding the cholesterol. So cholesterol is synthesis into the liver and go and deposit into the cell membrane, and water will be enter and through lipoprotein, and because the HDL, LDL, which that is the another topic we will not understand. So fluidity creation is basically endocytosis and cell membrane biosynthesis. So why cell membrane biosynthesis will be occur? Because the cholesterol gives the space where the space is occur, so the phospholipid will be attached. First bilayer of phospholipids will be again attachment and addition of that where the space is occur. So here we will discuss about the endocytosis and play important role of the fluidity due to the cholesterol. So here is the movement of the bacteria. For example, this cell is the macrophage. So when fluidity provide gap and clear, so it means the gap will be clear, a gap will be produced. It means the pinch inside of the plasma membrane due to the stretching, due to the fluidity is present. So that's form of fluidity and endosome will be formed, and again that cell membrane will cover their surface to complete. So this is known as the endosome, and this mechanism is known as the endocytosis. Endocytosis means the movement of material inside from the outside of the cell, while it is known as the endocytosed vesicle. So on the other hand, the exocytosis means the insulin, for example, from the beta cell of the pancreas will produce and become a vesicle. This vesicle is attached with the plasma membrane and export. So it means a little puncturing of the plasma membrane will release the insulin, and this insulin will go into the capillary. So remember about that. And this insulin will go into the capillary, so capillary of the blood vessel. So it means move inside of the blood vessel circulatory system, so where the need of the cell to bind this insulin to invaginate or invade the glucose due to the insulin binding to facilitate the glucose movement inside due to the glucose transporter channel. So this glucose transporter channel is the integral protein. So remember about that. So, and the next one thing we will discuss about the other thing, which that is the supportive mechanism of the plasma membrane and the cell cytoplasm. So the cytoskeleton is just like a human contain a skeleton, bone, and other cell. For example, the cellulose, as well as the basically the wood is used for the strength of the plant. Just like another things we will discuss in this diagram to understand. For example, so the important thing is that the plant support due to the wood. Human supporting mechanism, if we will talk about the physically, the bone, but inside of the cell there is the another mechanism to support the skeletal system. It is known as the cytoskeleton. Cyto means cell, skeleton means the structure or something which that provide the support. So here is the basically integral protein which that is attached with the collagen outside, and inside is the actin. Actin is used for the movement of cell. For example, amoeba is used for the actin and myosin. For the movement is streaming like movement. And on the other hand, if we will talk about the macrophage, which that is the same character in this living organism, is the eukaryote, multicellularity. So that is the evolutionary process. So stability, rigidity, shape of the cell, cytoskeleton composed with filaments which help remain in shape, and peripheral protein also which provide the rigidity as well as the provide the basically uh, structure. So on the other hand, enzyme containing protein also present, which that is basically, for example, enzyme containing protein is present in the cell membrane, in the digestive system, epithelial cell of the membrane, that is known as the enterocyte or brush border cell, which contain enzyme. This enzyme can be sucrase, peptidase, dipeptidase, which that is convert into the disaccharide into the monosaccharide, and dipeptide into the monopeptide. While on the other hand, here is the integral protein is known as the carrier protein specific. So this is the specific for every molecule and capture the molecule for movement inside by this change of their shape of the protein by the binding of the molecule or binding of the some stuff. So on the other hand, one more thing is a channel like protein is also known as the integral protein because it is embedded in the plasma membrane. So remember about that. Fluid mosaic model is the basically F Singer which provide the basically fluid mosaic model, which that is same like that, which provide the all information about that. So here is the channel containing protein, which that is sodium potassium exchange or something. So the hydrophilic and lipophobic ions, which that can be moved by the channel. On the other hand, the peripheral protein is used, for example, G protein, which that is used for the second messenger, and it is easily detached due to the hydrogen bonding attachment. Or on the other hand, it is non-covalent bonding, ionic bonding, which that is easily dissociate. On the other hand, peripheral proteins also contain a cytoskeleton, filament, tubulin, and microtubulin, and troponin, actin, myosin, which that is used for the strength and support and movement. On the other hand, the peripheral protein is example the G protein, and cytoskeleton is attached with the peripheral protein. Peripheral means beside of the cell membrane. So the cytoplasmic specific protein are present, such as transcriptional factor protein. These transcriptional factor protein is provide the second messenger. On the other hand, composition of membrane is the protein 50%, lipid is basically 47%, and carbohydrate is 3%. Of the 10% of that content, and other content 90% is the water. So I hope you make sense about that and the cell plasma membrane. Next we will discuss about the second messenger and the hydrophilic and hydrophobic interaction, and the second messenger and first messenger as a steroid. So thanks for watching.